Okay, let's do everything you ever wanted to know about the ovary. So the first thing to realize is the ovary has a hilum where blood vessels go in and out, and there's two different things happening in the ovary. There's like this amorphous stroma in which we are going to be finding follicles in the outer portion known as the ovarian cortex. And so we have tiny little inert follicles doing absolutely nothing, and then we have slightly larger follicles, and then we have even bigger follicles, and then you can see even bigger follicles over here. Then we have some follicles of varying sizes that have blood in them, including this really big one with all this big pink cells, which means it's producing lots of hormones. So let's kind of look at this and see what's going on. So we're going to have more than just the models from lab. We're actually going to have a lot of histology going on in this section. So what is a follicle first? Well, it's an oocyte, whether it's a primary oocyte or a secondary oocyte. It's an oocyte that's surrounded by follicular cells whose names are called granulosa cells. So it's the granulosa cells that are like the epithelial layer, and they're actually going to support the oocyte. So when the oocytes are hanging around for 20 years doing nothing, they really don't need much support from those granulosa cells. So they're very thin, almost not even seen here in the left lower quadrant, where you don't even see anything surrounding the oocyte. Whereas on other ones, you can clearly see a single layer, and then on some of them, you clearly can see two or more layers. Okay, so as they are developing. So hopefully from this picture, even though these are all like small, you can see that there are follicles in many phases of development at the same time. There is no follicle that starts on day 1 of the cycle and is ready to be ovulated on day 14. Okay, so it's going to take several cycles for a follicle to develop and be ready to undergo ovulation. And as it develops, the size of the follicle increases and the layers and number of cells increases, and the oocyte in it increases in size as well. So let's start at the beginning, pre-ovulation ovary, which has several steps. So the first ones, these are the follicles hanging out doing practically nothing. And these are called the primordial follicles. Over 99.9% of these are going to die. So you have, if you have one child, you have an heir, and you had many, many spares. And so you can see that these follicles are 99% just the oocyte. Now, some will stay as primordial follicles throughout your entire reproductive life, and some will progress and start developing. If they do start developing, they next become a primary follicle, and a primary follicle is going to be in meiosis 1. Every month, up to about 20 of them can start this process, probably closer to about a dozen. Anywhere between 9 and a dozen is normal. Some people have a few more, some people have a few less. You can tell these because when you look at the granulosa cells, you can see that there is one layer of flat cells. And it's actually in this stage at the zona pellucida that acellular layer is going to develop between the granulosa cells and the oocyte. And so the arrow is actually pointing to the cytoplasm of the oocyte, not to the zona pellucida. The zona pellucida is this skinny little line that you barely can see right there. All right. Now, what starts the process? Well, these hormones you're going to have hormonal stimulation coming from the hypothalamus and the adenohypophysis, and it's going to affect the granulosa cells. So initially, they're going to secrete a little bit, and it's a paracrine kind of thing where it stimulates nearby granulosa cells to start doing their things. And one of the things that happens is the granulosa cells, once they start being stimulated, they're going to start having an increase of receptors for follicle stimulating hormones. So when FSH is released by the adenohypophysis, there's more receptors, so more of it's going to bind. So then they're going to do even more stuff. So it's kind of like speeding up when you're falling down a hill. So this will continue to develop. That oocyte will grow to about 5 times its original size when it was a primordial follicle oocyte. And that zona pellucida becomes a little bit easier to see as this acellular layer. And this is what happens when you are in a secondary follicle, where you have more than one cell layer. It only takes 2 layers of granulosa cells to be a secondary follicle, but as you can see, we often have more. So this is one that only has about 2, and now you clearly can see the pink zona pellucida. So these multiple layers of cuboidal cells, as part of their work in making more estrogens, are also going to be secreting small amounts of fluids. And this actually starts about 2 months before a follicle will be ovulated, thereby giving credence to the fact it takes several months from the start of a follicle. If the follicle is starting in January, it's probably not going to be the follicle that's going to ovulate until April. Okay. So what we see as this secretes fluid, it starts separating the layers of the granulosa cells, and you can see that there's granulosa cells still attached to the zona pellucida, whereas other granulosa cells are still attached to the outside of the follicle. And eventually what happens is you end up with a mound of granulosa cells that covers the oocyte on one side of the follicle, and it actually kind of secures it to the follicle wall there, and the fluid collection kind of increases to the side of it. So how are these cells making their estrogen? Well, these stroma cells in the ovary absorb your cholesterol from the blood, and from that they make androgens. And I go, I know, androgens, testosterone, which is immediately almost converted, courtesy of your follicle stimulating hormones and your granulosa cells. They convert that androgen that's made into estrogen. So let's look closer at a secondary follicle, which is sometimes known as a mature follicle. This right there, we're going to do this question and answer, so don't move on until you know what it is. Yes, that's the oocyte. And you clearly can see its nucleus, and you clearly can see its plasma membrane. So what is this second arrow pointing to? Yes, that's the zona pellucida, the acellular area. Now, what's the third arrow pointing to? Yep, those are granulosa cells on the follicle wall. And this one, this particular cut, this big follicle doesn't show them attached, so I put another picture showing if you cut next to it, you can actually see that it's still attached there, and it's going to stay attached until ovulation. How about this fourth arrow? What is it pointing to? That's right, it's the antrum. And how about the fifth arrow? Yes, these are granulosa cells, but they have a special name in this location. Yeah. These are the cells, the granulosa cells that we call the corona radiata once we have ovulation. All right, so by the second week of follicle development, but no, I said that wrong. Not the second week of one follicle developing, but if we think of your menstrual cycle being once a month, so it's 4 weeks, by the second week of your menstrual cycle, one of your hundreds of developing follicles has become the dominant follicle. Other secondary follicles, which are really close to being dominant, like they're going to get the signal to undergo apoptosis and just become a scar. But one follicle becomes dominant while the others are degenerating because there's not enough FSH for everybody to go around. Yes. This is what happens. There's only food for the dominant one and the other ones are being starved to death. So this dominant follicle becomes the mature follicle, also known as the tertiary follicle. Do you like the way we go primary, secondary, tertiary? That's usually a little bit easier for students than all these other names. So the tertiary follicle starts bulging on the surface of the ovary, as you can see in this picture. It's eroding through the ovarian stromas. So do you notice all this tissue that was here on the secondary follicle? It's all been pushed away. And it's the tertiary follicle that's going to get the trigger, because it's still a primary oocyte. Yes, it was a primary oocyte way back here when it's a primordial follicle. And even though the follicle has evolved from primary to secondary to tertiary, the oocyte has stayed primary oocyte, primary oocyte, primary oocyte, and only the one mature follicle that wins the race is going to get the signal to complete meiosis 1. Hallelujah, and enter meiosis 2. So now finally we have a secondary oocyte. Only it gets stuck in meiosis 2, and it pauses in metaphase 2. And this only happens shortly, as in like a couple of hours before ovulation. So that brings me to something called the Graafian follicle, which is a subtype of the tertiary follicle. It's the tertiary follicle that's actually going to be ovulating. It is the one that has completed meiosis 1 and now is in metaphase 2 with a secondary oocyte. So we have this huge follicular antrum, which accounts for like 95% of this follicle. And you can see it's really eroded through a lot of the ovarian tissue, but it still has to kind of work its way through that. So at the time of ovulation, this entire package is what is released, and only this package. So you're going to have three things. You're going to have our secondary oocyte, which is paused in meiosis 2, everything lined up in metaphase 2. You're going to have the secondary pellucida and on the zona pellucida, and on the outer part of that, you're going to have the corona radiata. So at ovulation, hallelujah, you can get pregnant if Mr. Spermie is around. Now, during your normal lifetime, only 1% of the gametes you're born with will ever be ovulated. We'll be showing you more details about that later on. So this on your cartoon drawing is the picture of ovulation, because looky, it is free out there. And here is a gross picture of, gross as in you see with your eyes, not gross as in, ooh, it's gross, but gross anatomy picture, where you can see the hemorrhage at the site of ovulation. The follicle that ovulates is hemorrhagic. So this actually was a random occurrence where someone was having a laparoscopic surgery for something else, and the physician was astute enough to realize that ovulation was occurring at that very moment, and took pictures showing ovulation. This process is amazing process. All right. So what happens to the ovary after ovulation? Well, pre-ovulation, we have the number of cells increasing, the granulosa cells, you know, they had FSH receptors, so the estrogen levels were going up and up. And after ovulation, some granulosa cells left with the oocyte, but the vast majority stayed behind. And now they're thinking, she might be getting pregnant courtesy of Mr. Sperm. So this becomes a steroid hormone factory, and it starts producing massive amounts of progesterone in anticipation of the pregnancy. Yes, it also secretes estrogen. But what the progesterone does is it gets the endometrium ready for implantation of the fertilized ovum. And you're going, well, no, isn't that the job of the placenta? Dude, there's no placenta. Placenta doesn't start developing for weeks and will not maintain the pregnancy until pretty close to the end of the first trimester. It starts developing, 5-6 weeks into it, but it's not capable of maintaining the pregnancy. It's this these granulosa cells, which have become this progesterone factor, and remember, progesterone is a lipid hormone. Lipids are fats, thereby it turns bright yellow in color, and we now call it a corpus luteum or corpus luteum. Now, it's going to do that. It's going to crank out massive amounts of hormones for about 10 days. And meanwhile, it keeps on asking, is she pregnant yet? Is she pregnant yet? Is she pregnant yet? Because what it's looking for is if fertilization occurred, then what happens is that developing embryo starts kicking out a hormone, which is HCG, human chorionic gonadotropin. I'll talk about it a little bit more later. And this will give positive feedback for the corpus luteum to continue, make even more. And what happens is this corpus luteum actually gets 2 to 3 times larger than the size of the pre-ovulation ovary. It's huge in size. Okay, it's huge in size even without getting pregnant, and then it just gets super huge inside if you are pregnant. Now, if within 10 days it doesn't get any HCG signals coming back, then it goes, I don't know why the hell I was working so hard. We're going on strike. It is siesta time for another month, and it will degenerate. So here is a fresh surgical ovarian specimen where you clearly can see a fairly significant corpus luteum that still has blood in it. So this is not that long after ovulation. This is going to be within a couple of days of ovulation. Now here is a earlier corpus luteum that has started degenerating. Notice how much smaller it is. Okay, so when I say it degenerates, it doesn't go away in a week like a little cut on you. It can take a significant amount of time. And I think on this, you also kind of get the idea that there's probably another old one in here. And so because this corpus luteum, it takes up so much of the ovary and the resources for that ovary, this also lends credence to the fact that most women will ovulate on one ovary 1 month and the other ovary the subsequent month, and they alternate back and forth between the two. So this is a recent corpus luteum, so this one has to be at least 2 months old. So when the corpus luteum degenerates, it is going to turn into a little white scar known as the corpus albicans, because ALB means white. And you clearly can see a little corpus albicans action going there, and there's a little bit going on there. And then this is a fixed surgical specimen where you can see this is not a brand new corpus luteum because there's no hemorrhage left in it. So this is an older corpus luteum, and then you can clearly see 2 other corpus luteum from before, and you can see lots of corpora albicans in there. So let's look at this from a histology part. So in order to identify a histology on corpus luteum, you are looking for something that you can see is circular, so it's a follicle, and it is chock full of cells. You see all these cells, which are paler in nature because the lipid is accumulating in there, all that progesterone it is making. And these cells have massively proliferated, which kind of gives it this undulating appearance. Because it's filling up that space in the antrum, and the cells aren't just growing like this, they are growing as if, you know, like in the brain where you have the gyri of the cell socket. Okay. So that's what a corpus luteum look like. And then a corpus albicans, you just are going to have a small squiggly white scar. Here is an early corpus albicans because it's quite a bit larger. But I think if you look at this up close, you pause it, you would see a distinct lack of nuclei in this structure. And in this structure, because it's a star, it's mostly collagen. Whereas in these, there are lots of nuclei because these are cells actively producing hormones. All right. So it's time for a DWAP. There are cartoon drawings on this side, so talk yourself through the process of what is happening with follicular development, and also don't forget to talk about hormones there as well. And now here there is a second GWAP, which has to do with histology slides. So see what you can identify. There are lots of structures to identify here. And with that, we are done with our exploration of the ovarian structures, and we're going to do a little bit more talking about the ovary in detail without pointing out the structures. So see you again shortly.